Good afternoon, students. Today we are going to discuss with intergenic separation, also called as extragenic separation. In the last lecture we discussed about genetic separation. What is genetic separation? What is true reversion or back mutation, and then we studied about genetic separation, wherein we studied intragenic separation, where the suppressor mutation or the second mutation takes place in the same gene where the first mutation has taken place, and it suppresses the effect of first mutation. Now let us discuss about intergenic separation or extragenic separation. Intergenic separation is that where the suppression of first mutation in one gene is brought about by second mutation, also called as suppressor mutation, in another gene. That means the two mutational events take place on two different genes. Right. So intergenic separation or extragenic separation is that where the second mutation or the suppressor mutation takes place in another gene. Okay. Now the second mutation that suppresses the deleterious effect of first mutation takes place on other gene. The two genes may even be present on different chromosomes. Okay. And how this extragenic or intergenic separation is achieved? It can be achieved mainly due to alteration in anticodon of tRNA. Okay. So extragenic separation can be achieved by alteration in anticodon of tRNA, where the anticodon of tRNA is changed. How? How it suppresses the effect. Now see, there are nonsense mutations. Okay, we have discussed it earlier. What is a nonsense mutation? Nonsense mutation is the one in which a sense codon is converted into a nonsense codon, which may be UAA, UAG or UGA. Because a sense codon is converted into a nonsense codon is not read by any anticodon of the tRNA, and hence the protein synthesis abruptly stops. Okay, so premature chain termination takes place, and the polypeptide which is formed is shorter in length and most of the time partially or completely non-functional. Okay. So how to suppress the effect of this nonsense mutation? The effect of nonsense mutation can be suppressed by altering the anticodon of tRNA. How it is brought about? So it is altering the anticodon of tRNA is one of the method for suppressing the effect of nonsense mutation. How it happens? See, in nonsense mutation, a sense codon gets converted into a nonsense codon. Now, the nonsense codon cannot be read by any anticodon of tRNA because such an anticodon does not exist. Okay. So, what is done in genetic separation? The anticodon of tRNA is altered in such a way that, okay, the anticodon of the tRNA is altered in such a way that it can pair with termination or nonsense codon. Okay, so the anticodon of the tRNA is changed such that it can now pair or read even the nonsense termination codon and add a amino acid. Okay, and amino acid is added and protein synthesis continues, and the protein may regain its functionality either completely or partially. Okay, so altering the anticodon of tRNA will suppress the effect of nonsense mutation. Now let us discuss some examples. Now one of the codon for glutamine is CAG. If you go from 5' to 3' end, it is CAG. Now this codon is recognized because it is a sense codon. It is a codon of glutamine. Okay, so this codon is recognized by the anticodon GUC. Okay, this is the anticodon of glutamine. So, if the codon present on mRNA is CAG, it is glutamine codon. It will pair with the anticodon of the tRNA, that is GUC, and glutamine amino acid will be added into the protein chain. Because of nonsense mutation, the glutamine codon CAG is converted into UAG. Okay, so the moment C is converted into U, CAG gets converted into UAG. UAG is an amber termination codon. It brings about termination of protein synthesis. The polypeptide chain synthesis abruptly stops. The incomplete polypeptide chain formed is usually inactive. Okay, got it? So CAG converted into UAG because UAG is a termination codon, protein synthesis will stop. Incomplete protein formed is inactive. Okay. Now, how the effect of this nonsense mutation can be suppressed? The effect of this nonsense mutation can be suppressed by mutation in another gene. And where the anticodon. Okay. The normal anticodon of tyrosine tRNA. The normal anticodon which is present on tyrosine tRNA is AUG from 3 to 5 prime end. It is AUG. So anticodon of tyrosine tRNA is AUG. AUG pairs with what? Okay. Okay. Now a suppressor mutation converts the anticodon AUG into AUC. Okay, that means G to C transition takes place. Right? The normal codon for tyrosine tRNA is it is actually the anticodon. Normal anticodon of tyrosine tRNA is AUG. Suppressor mutation which converts G to C will convert AUG anticodon into AUC anticodon. Now this AUC is mutated tyrosine tRNA. This is tyrosine tRNA. It is going to add tyrosine amino acid, but it is mutated. Originally it is AUG. Because of suppressor mutation, it has been converted into AUC. Now this mutated tyrosine tRNA having the anticodon AUC reads the termination codon UAG. It can pair with the termination codon UAG and add tyrosine in the protein chain instead of glutamine. Okay, so instead of glutamine, where originally in the wild type chain there was glutamine, now because of mutation it was converted into nonsense mutation and polypeptide chain synthesis stopped. So alteration was done in the tyrosine tRNA such that the mutated tyrosine tRNA reads the termination codon and tyrosine amino acid is added in place of glutamine, and rest of all amino acid will be same. Okay, so the mutant protein regains activity. In this case, the suppressor gene functions by producing tRNA that can read a termination codon. Okay, so this is how the deleterious effect of a nonsense mutation can be suppressed by altering the anticodon of tRNA such that the mutated tRNA can even read the termination codon and carry out the protein synthesis. Okay, let us look at this. This is a tRNA of tyrosine. Okay, so it's a tyrosine tRNA having anticodon AUG. You can see here the anticodon of tyrosine tRNA is AUG. Okay, now this is your mutated mRNA. Okay, because mutation has taken place, CAG has been converted into UAG. So this is a nonsense, you can say mutant. One minute. Yeah. So this is a nonsense stop codon. Okay, in presenting mRNA, CAG has been converted into UAG. Can UAG be read by any anticodon? No. So what is done here is AUG, which is the normal anticodon of tyrosine tRNA, undergoes suppressor mutation whereby it is converted into AUC. This AUC is the anticodon of mutated tyrosine tRNA, altered codon. Now this AUC can pair with the termination codon UAG and tyrosine amino acid will be added to the growing polypeptide chain, and thereafter the protein synthesis will continue. This is how a nonsense mutation is suppressed. Okay. Now let us take another example. A nonsense mutation has taken place in the gene such that the codon present on the mRNA, normal codon is UUG, it pairs with AAC and the amino acid leucine is added. Now because of nonsense mutation, the codon UUG has been converted into UAG. Okay, because of nonsense mutation, the codon UUG is converted into UAG. Now UAG is a stop codon, it's a termination codon. So translation, protein synthesis will stop. Now, how the deleterious effect of this nonsense mutation can be suppressed? So, now a nonsense suppressor tRNA. What is done? Right? Now, this is the tRNA, wild type tRNA for tyrosine. This is tyrosine tRNA having the anticodon AUG. So, it's a tyrosine tRNA having anticodon AUG. What is done here during suppression? The AUG is converted into AUC. This AUC can pair with the nonsense codon UAG, and the mutated tyrosine tRNA will add tyrosine in place of leucine in the growing polypeptide chain, making the protein functional. Okay, this is how effect of nonsense mutations are suppressed by altering the anticodon of the tRNA. Now, we also know that along with nonsense mutation, missense mutation also occur, frameshift mutation also occur. So, missense mutations are also suppressed. Missense suppression and mutant tRNA. These mutations will lead to synthesis of a protein. What missense mutation does? Because of missense mutation, a protein formed has only one amino acid different. Okay, only one of the amino acid is changed at a particular site. Such a mutation can also be suppressed like that of nonsense mutation. How? By using a mutant tRNA, which will misread the codon and substitute a correct amino acid. Okay. See, why there is a change in a single amino acid in the protein? Because the codon present on the mRNA is mutated. Okay, the gene has undergone mutation. So, how the effect of this missense mutation can be suppressed? By using a mutant tRNA. Now, what is the mutant tRNA doing here? The mutant tRNA will misread the codon and substitute or add a correct amino acid, although the mutant codon codes for a different amino acid. Okay, say for example, A36 in tryptophan synthetase of E. coli, tryptophan synthetase gene of E. coli. The glycine which is coded by GGA is replaced by arginine. We have studied this, which is coded by AGA. The suppressor mutation in tRNA will deposit glycine for the codon AGA, which is actually meant for arginine. That means though the codon has changed from GGA to AGA, and AGA codes for arginine because AGA is read by its complementary tRNA. Now the anticodon of the tRNA is changed in such a way that it will misread the codon AGA, and instead of adding arginine, glycine will be added in its position, and the effect of missense mutation can be corrected. Okay. So, this is the anticodon, glycine codon, okay, GGA. Because of missense mutation, some change has taken place so that arginine can be added, but the missense mutation can be suppressed. Suppression mutations involving a change in one base of the anticodon of glycine tRNA, leading to misreading of arginine codon AGA, which resulted due to missense mutation in glycine codon and leads to the suppression. So, the effect of missense mutation can be also suppressed by altering the anticodon, or by bringing about a suppressor mutation such that the anticodon will misread and add the correct amino acid instead of the wrong amino acid. Similarly, frameshift mutations are also suppressed. Here, what happens in a frameshift mutation? Because of addition or deletion of a base, the reading frame is shifted either in backward direction or in forward direction. That means left side, towards the left if there is addition of base, or towards the right if there is deletion of base. It has been shown that frameshift mutation because of deletion can be suppressed by a mutant tRNA having an anticodon containing only 2 bases rather than 3. Okay, so if one of the base is deleted from a codon, then that effect of that deletion mutation can be suppressed by a mutant tRNA which is having only 2 bases in its anticodon. That is called as doublet anticodon rather than triplet. Similarly, frameshift mutations which occur because of addition of a base, okay, can be suppressed by a mutant tRNA which is having a quadruplet anticodon. Okay, instead of a triplet, that means which is having 4 bases in the anticodon, and this is how the effect of frame shift mutations can be suppressed. So mutant tRNA with doublet and quadruplet anticodons have actually been detected in biological systems which can suppress the effect of frame shift mutation. So whether it is nonsense mutation or any other mutation whose effect is suppressed, or whenever genetic suppression takes place in an organism, it is because of the suppressor genes which bring about the genetic suppression. So let us discuss in brief about suppressor genes. So what are suppressor genes? Genes whose activity suppresses the effect of mutation in another gene. Okay, gene whose activity suppresses the effect of mutation in another gene are called as suppressor genes. So the suppressor genes function by suppressing the effect of mutations. Okay, primary mutations. There are suppressor gene for all the 3 termination codons. We have 3 termination codons, UAA, UAG and UGA, that is ochre, amber and opal. Okay, so suppressor genes exist for all the 3 termination codons, right? If you look at amber suppressor genes, there are 3 suppressor genes for amber anticodon and amber termination codon. What are those? SU1, SU2, SU3, also called as SUPD, sub D, sub E and sub F. So these are the 3 suppressor gene which can suppress the effect of nonsense mutation which occur because of amber termination codon, that is UAG. And how do these suppressor genes act, or how do they bring about the genetic suppression? They bring about genetic suppression by adding either serine or glycine or tyrosine in the termination position. Wherever premature termination of polypeptide chain synthesis has taken place because of nonsense mutation, the suppressor gene will alter the anticodon of tRNA such that either serine or glycine or tyrosine is added in the termination position. Similarly, there are 2 suppressor genes which suppress the effect of ochre termination codon. They are SUPA, sub A and sub B. Then opal suppressor gene also exists which suppress the effect of nonsense mutation which occur because of opal termination codon, that is UGA. Now the nonsense mutations which occur because of UGA are suppressed by adding tryptophan amino acid in the termination position. Okay, every time the tyrosine the anticodon of tryptophan tRNA is altered such that the mutated tryptophan tRNA will add tryptophan in the termination position, the termination which has occurred because of UGA. Okay, so suppressor genes do exist for all the 3 termination codon which help in suppressing the effect of nonsense mutation. If we look at the you can say percentage or frequency at which the suppression of nonsense mutation is brought about, suppression of UAG and UGA is about 50%, while the suppression of UAA termination codon is less efficient and is only 1 to 5%. So this is how suppressor gene help in suppressing the effect of nonsense mutation. So that is all about genetic suppression.